Hello to everyone and thank you for joining. My name is Alja Videtič Paska and I will present you our work on Alzheimer's disease that is a result of collaboration between Faculty of Medicine in Ljubljana and Institute Ruđer Bošković and University Psychiatric Hospital Vrapče, both in Zagreb, Croatia. I will just start with a few facts about Alzheimer's disease that you probably already know, but that still startle me every time I hear them. So today there is more than 40 million people worldwide living with Alzheimer's or another type of dementia. By 2050, this number will reach 110 million people worldwide. This means that 1 in 3 seniors dies with Alzheimer's or another dementia. However, Alzheimer's disease does not start overnight. We know that the changes in the brain start more than 20 years before the onset of the first symptoms. Unfortunately, the problem of Alzheimer's disease is still the diagnosis, which is established too late, which reduces the effectiveness of currently existing pharmacological therapy. This is why we are still on the lookout for reliable biomarkers, which could be used to early and accurately diagnose Alzheimer's disease. This brings us to the focus of our interest, brain-derived neurotrophic factor, or BDNF, and catechol O-methyltransferase, or COMT, and the potential role of different methylation patterns of these genes in the development of Alzheimer's disease. One of the most frequently investigated genes associated with cognitive phenotype is the gene for COMT, an enzyme that degrades catecholamines. In terms of the onset of cognitive decline in dementia, the activity and expression of COMT is especially important in the prefrontal cortex, the brain region, which is engaged in various cognitive processes and regulated by dopamine. Therefore, COMT has been studied as a target for different neuropsychiatric disorders and cognitive impairments that have been associated with altered dopamine function. As for the association of COMT gene with dementia, most research to date has focused on COMT gene polymorphisms rather than its expression and activity. In addition, another risk gene, the BDNF, is also associated with cognitive impairments in various states in health and disease, including Alzheimer's disease. BDNF affects the growth and development of neurons, synaptic plasticity, and neurotransmission moderated by different neurotransmitters, cognition, learning, and memory. So far, the reduction of BDNF concentration in different brain areas has been associated with dementia and cognitive decline. In our study, we collected more than 250 blood samples of subjects with mild cognitive impairment and Alzheimer's disease. We applied contemporary methods like next-generation sequencing and determined methylation status of Alzheimer's disease genes, COMT and BDNF, in white blood cells in clinically well-defined Alzheimer's disease patients and subjects with mild cognitive impairment. Subjects with mild cognitive impairment were used as a comparative group since we had a lot of problems finding control subjects that were old enough and without cognitive disturbances. The project also aimed to determine BDNF and COMT methylation on circulating cell-free DNA from plasma, and also gene expression levels and specific single nucleotide polymorphisms that could be associated with BDNF and COMT expression levels. We have now finished most of the analysis planned in the project, but we still have to process the data and then compare the results. This is why we will focus today only on BDNF and COMT methylation status. Using PCR amplicon library preparation approach, we sequenced 9 amplicons residing in BDNF gene covering 169 CpG sites and 4 amplicons residing in COMT gene covering 62 CpG sites. Changes in methylation pattern between mild cognitive impairment and Alzheimer's disease groups were observed for both genes. Biggest change was observed in the 2 BDNF amplicons located at the CPG islands in the promoter region and in the first intron of the BDNF gene. On average, we observed lower levels of methylation in both BDNF amplicons in patients with Alzheimer's disease compared to subjects with mild cognitive impairment. We hope to confirm these results in cell-free DNA and by exploring the expression of BDNF and COMT in white blood cells. But even these preliminary results suggest that exploring epigenetic markers of Alzheimer's disease should not be limited only to the central nervous system, since the periphery can also give us some interesting answers and broaden our knowledge. Sometime in the future, they might be used as potential epigenetic biomarkers for predicting the progression from amnestic mild cognitive impairment to Alzheimer's disease. In the end, I would very much like to thank all the co-workers that collaborated in this work, the funders, of course, and the organizers for giving us the opportunity to present our results. Thank you.